to turn the recording on. There we go. Y'all get a little message that says, hey, you're getting recorded. If you're not okay with that, all that kind of good stuff. Well, thanks everybody for turning out tonight. I'm excited to get the full presentation. I've up to this point had just little snippets of Lisa's work, so I'm really excited to get the full meal deal, so to speak, because fascinating stuff, great stories. Some of my favorite stories come out of some of the results of your research, so I'm excited for folks to hear some of that cool stuff tonight. Do mute yourself during the program. If you want to throw questions in the chat box, you can certainly do that. We will hold off on any questions till the end of the presentation, but you can certainly, like I say, pop questions in the chat box. And if it's something that looks like it's really urgent that it needs to be dealt with right now, I'll pop in and politely interrupt and let Lisa know so she can answer that. But most of the time that's not an issue. We don't need to worry about that. Yeah, thanks for coming in out of the lovely weather and being ready for adventures tonight. Lisa, let's see, you finished your master's work. Now it's going to be two summers ago, yes?
Well, fall 2020 was when I defended my thesis.
Okay, so wow,
yeah, well, over 2 years now, yeah.
More than 2 years, but still one of the most fun projects and one of the coolest research projects I think that has been done. So I'm excited to have Lisa give us the rundown tonight so that you all can hear that too, and so we can record it and keep it around in posterity. So yay. All righty, thanks Lisa. I am going to make you the host so you can do your thing. There we go. Still got people popping in, but now Lisa's the host, so she can go ahead and share. And
now I have to be like, does everyone see the screen? Alrighty. Well, thank you, Margie. I've been away from Duluth for a couple years now, but I miss it dearly. So it's always fun to be able to, kind of pop back and talk with y'all. So today I'm going to talk about the work I did for my master's thesis at UMD. And I used DNA metabarcoding to study raptor diet during migration at Cockridge. So today I'm just going to kind of go over my thesis. We'll go through the background information, talk about, why dietary studies are important and why, we had this need to develop a new method to study diet in migrating raptors. A lot of research has been done on raptor diet, but there's a big bias in those studies being done during the breeding season, mostly. Some studies that are being done during, overwintering habitats, but there's this real knowledge gap in migratory diet studies. So go over a little background, talk about what DNA metabarcoding is. I'll walk through the methods and just results, discussion, and conclusions. All right, so we'll start off with, why is it important to study diet? The first thing is that it's a very essential part of just understanding the basic ecology of these birds. So, we might see a seasonal effect on diet. So, for example, broad-winged hawks, when they're, breeding in northern Minnesota in June, you might imagine that's a very different prey availability than when they're overwintering, in the tropical forests of Panama in January. So that's kind of a dramatic scale, but even, on a smaller scale, how does prey abundance vary, from like late May until August in Minnesota? And does, the prey abundance affect, what they're eating at different times of the year? So then we also have this question of like, is there differences in diet between ages of raptors or the sex of raptors? we got the classic example here of Cooper's hawk. You have the female Cooper hawk on the left and the male on the right, and she's about a third bigger than her little male buddy. And so one of the theories about why we might see sexual dimorphism in raptors is that it reduces competition for food between the sexes. So perhaps the female is hunting larger prey, and maybe the male is hunting, slightly smaller but harder to catch prey. So he has that like little boosted agility in there. So it's a great hypothesis, but up until this point, there really hasn't been enough data to say, whether there is a significant difference in the diet between males and females or adults and juveniles in raptors. And then dietary studies have big implications on conservation. So for example, land management, places like Duluth, we want to be providing good raptor habitat at Hawk Ridge. And so if you provide habitat for the prey, the raptors will follow. And then on the opposite side of that, though, places like airports, they actively want to deter raptors, right, to prevent bird strikes. So that's what this paper in the lower right-hand corner did. They analyzed the stomach contents of birds that were hit and killed by airplanes, so that they could then inform, the airport management, like, how they could change their landscaping to lower the prey abundance and other food. And we can't talk about conservation without talking about climate change. It's definitely the biggest conservation concern for most species now. So we know Minnesota is getting warmer, and we know that it's getting wetter, and this is going to change our habitat in Minnesota. And so how is the changing habitat going to affect the prey abundances and what raptors are feeding on in our area? And we really can't say whether or not there's change over time unless we have a really good understanding of what the diet was to begin with. And as I'm sure most of you know, like, there's a lot of environmental contaminants that raptors are exposed to through their diet. So classic examples like DDT, or the example on the right here is mercury. So by understanding, what raptors are feeding on, we can like really assess, well, what's their exposure risk to some of these contaminants. And finally, there's still a lot of work to be done out there with like public awareness and education. Photo on the top right is from Hawk Mountain in Pennsylvania. That's just one of a few of the hundreds, maybe thousands of hawks that were shot and killed there because they were viewed as pests, right? They're going to eat your chickens, they're going to eat your grouse that you want to hunt, so we got to kill them first. And thankfully, this isn't as big of a problem in the United States anymore, but globally, raptor persecution is still a major conservation concern. So anything we can get out there to do, to talk with the public and dispel misconceptions about what raptors are eating, has a real impact. All right, so I'm just going to go over quickly some of the common methods that have previously been used to study diet in raptors. So first off, we've got stomach content analysis. This was the most popular method in the late 19th, early 20th century. You just go out and you shoot the raptor and you cut it open, you see what's in its stomach. So obvious drawbacks to this method is, you've either got to kill the bird or you have to find a dead bird. And there are still studies that use stomach content analysis, like that airport study I talked about a minute ago. But obviously not really the option that I wanted to do for my research. And then there's pellet analysis. This is a very popular method for studying diet in owls. And the traditional method of using pellets was a morphological analysis of what was in there. You have to be a very good expert on mouse bones to do that. But nowadays we also can use DNA and molecular methods to analyze the contents of pellets. Of course, there's nesting studies. one of the most common types of dietary studies you'll see in the scientific literature. You know, a couple different ways people do this, you know, collecting prey remains at the nest, setting up a camera. It's all great methods, but it's highly biased to the breeding season diet. And then there's good old direct observation. You know, it's not easy. It's very time consuming. Obviously, you know, very hard to do with nocturnal species, and, you know, you might not get a very precise prey identification. All right, so those were the old methods. So onto the new kid on the block, which is DNA metabarcoding. DNA metabarcoding is just a molecular technique that's used to identify DNA in samples with DNA from a lot of different species in that sample. So it does this by IDing a unique barcode for each species. And then those barcodes are sequenced with high throughput sequencing. You'll sometimes also hear this referred to as like second generation sequencing. The old sequencing technology could only sequence like one DNA sequence kind of at a time, right? So if you had DNA from two different species in it, it just couldn't handle that. It would have to, you know, do the turtle DNA first and then do the grasshopper DNA like in separate runs. High throughput sequencing, it can spit out the results all in one. And DNA metabarcoding got its start in the microbiology world, you know, using to study, you know, microbiomes and samples with many different species of bacteria in it. It's nowadays really commonly used in environmental surveys to identify species from water samples and soil samples. So it's, you know, used to both like monitor and detect, you know, aquatic invasive species, but also to survey for, you know, threatened, endangered, or otherwise like rare species, because all you have to do is scoop up a little water. And then, of course, dietary studies. All right, so for dietary studies, one of the most frequently used sources of material for the dietary DNA is feces. If you guys are familiar with raptors, that you may know that it's quite difficult to obtain raptor feces. They don't poop as much as other bird species. So for, you know, songbirds, DNA metabarcoding with fecal samples is, you know, fairly frequently done because all you need is a sterile bag or a box, and you put your bird in there, and, you know, 15 minutes later you check the bag and you've got your fecal sample. But can't do this with raptors, so we had to think up of another method that we could use to collect fecal material. So we decided to use cloacal swabbing. So as a little like background on bird anatomy, the cloaca is the orifice that birds have for both feces, nitrogenous waste, and reproduction, so all in one. Previously, cloacal swabbing has been used to detect diseases like West Nile virus and avian flu, study microbiomes in birds, and then also as just a, we'll say, semi-non-invasive way to collect genetic material from the bird itself. And then when I did this project, it had not yet been used to study diet. So yeah. For this project now, we had like kind of two basic hypotheses, questions we really wanted to answer was, first of all, whether or not we could even use cloacal swabbing to obtain, you know, dietary DNA or prey DNA from raptors. And then our second hypothesis was like, okay, well, if we do get the dietary DNA, is the detection of dietary DNA going to vary between different species of raptors? So first hypothesis, you know, like, of course, I was hoping that, you know, one of two outcomes, like if we would detect the dietary DNA and I would be a very happy master's student. But, you know, no one had done this before, so we knew there was a real possibility that we might not detect dietary DNA for reasons like, you know, raptors might not be feeding very frequently during migration. If they're not feeding frequently, they're not pooping very frequently. We also thought, we don't really even know how much fecal residue hangs out in the cloaca. And then what quality is that DNA from the prey, once we get it on the swab, because it's gone through the digestive system, it's, been hanging out there for a while. The second hypothesis was, again, that the amount of dietary DNA detected would vary between species. In the first way I saw this as, happening is that there's a big difference in physical size between these raptors, right? Large raptors, bigger cloacas, maybe more fecal material, and then just more DNA to work with, as opposed to our small raptors, smaller cloacas. And then we were thinking, well, maybe they'll have less dietary DNA. And then the other way that we thought that thing that might have an effect between species was, how these raptors are migrating during migration. Are they, actively flying like sharp-shinned hawks or kestrels, lots of flapping involved. It's a more energetically expensive way to migrate. So we're thinking, hey, maybe these birds have to feed more frequently. They'd feed more frequently, poop more frequently, and so on. And then on the other hand, raptors like red-tailed hawks, broad-winged hawks that are primarily soaring during migration, they don't need to use as much energy, so maybe they're not going to feed as frequently, and they would have less dietary DNA detected. All right, that was the hypotheses. I'm going to kind of just quickly walk through the methodology here. First up was the most fun part, which was sample collection. I collected my samples during 2019. We did a preliminary sample collection in the spring just to make sure that it worked, or we thought it would work, and we swabbed 81 raptors in late April, early May, and then the bulk of the sample collection was in fall of 2019 during the migration, August to November. I've got my samples there in the table on the right. That includes both spring and fall for those totals. So I collected from 11 different species. I'm sure the first thing you'll notice is that the number of samples I collected between species really varied greatly, from the one little rough-legged hawk I swabbed up into the 141 sharp-shinned hawks. And we just decided that, it was better to prioritize collecting the most swabs from the widest variety of raptors, just because, again, we hadn't done it before. We didn't know what kind of variation we would see between the species. So then fast forward a couple months, those swabs hang out in the freezer until we're ready to extract the DNA. After the DNA is extracted, then we amplify the barcode, the identifier we're interested in, using PCR. And this is the same, molecular methodology that, you hear about with COVID testing with PCR. And all PCR does is it takes DNA that's in a very low concentration and it amplifies it. It makes more physical copies of those DNA sequences so that you have enough material to run it through a sequencer. So for this project, we decided to target barcodes that focused on avian prey and mammalian prey. So these were regions of, if you're into genetics, it's the 12S mitochondrial gene. And it was 160 base pair long segment. So fairly short, because the shorter the DNA sequence you pick out when you're dealing with dietary DNA, the higher likelihood you have of that sequence being intact, because again, the DNA you're sampling has gone through the digestive tract of a bird and has been broken down quite a bit. So if you tried to sequence something that's a longer sequence, you might not have that full sequence intact. So PCR, we've got our amplified DNA. Then we send it off to another lab that does the high throughput sequencing. And then that spits out millions upon millions of sequence reads. And so the bioinformatics step then sorts those sequences into ones that are the same, and those are called operational taxonomic units, or OTUs for short. And for today, like, just think of OTU as a proxy for species. It just means, it was all the same DNA sequence. That's not like, it's not the same as species, because if you had two very closely related species and they happen to have the same DNA sequence for the barcode we were looking at, then they're going to get lumped together as the same OTU. And likewise, if we had, two individuals of a species that has a lot of genetic variation, if those two individuals had different DNA sequences for our barcode, then they would get lumped into separate OTUs. But like, we picked our barcodes because those regions had been shown to be unique between species, but have a lot of consistency within a species. All right, so then once we've got all of our sequences sorted into the OTUs, then we run those sequences through a database of sequences and see if we have a match for a species. Okay, so take a little sip here. So I'm going to go through the results now. Little drum roll. We found that 18% of swabs had DNA from at least one prey species detected on it. So of those 53 swabs that had dietary DNA detected on it, the majority had only one prey species detected on the swab. 8 swabs had 2 prey OTUs detected on it, and only 2 of those swabs had more than 3 or more prey species detected on it. And of the 11 raptors that we collected samples from, these 5 had dietary DNA detected. I think the main reason we didn't get detections on the other species is just due to like such a small sample size. But what I really like about this table here, it's each bar is the total number of swabs for that raptor that had prey DNA detected on it. And then the colors signify whether that prey DNA came from avian prey in blue, or mammalian prey in red. And then in one instance where in the red-tailed hawk, where the swab had 2 different species on the swab, where it was both bird and mammal. And what I love about this is it's exactly what we would expect, right? Sharp-shinned hawks are mostly eating birds. Cooper's hawks, northern goshawks, kind of a mix of mammalian and avian prey. And then you've got, red tails and sawets with mostly their mammalian prey. So that was good, because I'm not trying to argue here that these raptors aren't eating what we're mostly expecting them to eat. So now I'd like to look a little bit more closely at like what those OTUs were. We had 35 unique prey OTUs identified on the swabs. Of those 35, we were able to have a close to perfect match for 19 OTUs. That's not abnormal, and as I'll talk about in a minute, like, not every species is in the database. So it's not that those other 16 prey OTUs weren't real detections. It's just we had to assign them to a higher taxonomic level. Like, we could say, oh, this OTU came from a sparrow, but we're just not sure what species of sparrow. And as far as like the breakdown of types of prey, we identified, 23 came from birds and 12 came from mammals. So after I got my data back and was a little sad that I wasn't able to identify 16 of these OTUs, I was kind of curious, like, well, how many species are actually in the database? So I did a little post hoc search of the database where I just, manually searched a bunch of species in our region. And I found that, about 54% of birds and 30% of mammals, when I was doing this in 2020, had not yet had the 12S gene, the gene where our barcode is located, sequenced. And of the ones that had the 12S gene sequence, it wasn't necessarily fully sequenced. So that could also explain why our barcodes weren't having matches. All right, so I can't walk through all 53 swabs, but I did want to take a minute to just talk about northern saw-whet owls to kind of explain the data a little bit more. Of the 22 saw-whet owl samples we collected, 7 of them had prey species detected on them. So each line here is an OTU. So we have listed the closest match for the OTU in the center column, and then what percentage similarity that sequence was to, for the first line, the deer mouse. So anything with, like a 98%, we're fairly confident it's a mouse, just maybe not what kind of mouse. The northern short-tailed shrew here, that's 99% similar. Maybe that's within species variation for short-tailed shrews. But the bulls, especially, as I found out later, have very complicated genetics. So we're confident they're bulls, but we just don't know what type or what genus. So those are kind of expected findings. I'm going to go through some of the fun things we found. My favorite was definitely a male juvenile goshawk that was swabbed on the 23rd of October and had Virginia opossum DNA on it. That sequence was a perfect match, so we are highly confident it ate an opossum. And if you guys are from Duluth or you're familiar with Duluth, there aren't opossums in Duluth, or at least they're not very common at all. You've got to drive about 60 miles south of Duluth on 35 before you start seeing dead opossums on the side of the road. And opossums weren't even in Minnesota, oh gosh, 100 years ago. They've spread into the state, and since then they've been moving northward. So yeah, when I saw this, I was thinking okay, did this goshawk just happen to eat the one opossum that's in Duluth or north of here? Or had it actually come from the south and flown northwards after eating an opossum down by Hinckley? So I can't say what happened, but to me, this is just a really good example of how studying diet can tell you more than just what a bird is eating. Maybe it's a way to help detect rare species on the landscape, or maybe it tells us something more about migration patterns for raptors. And then as another little side note, all I can say is the goshawk ate an opossum. I don't know if this was predation or if this was scavenging. But yeah, it's just fascinating. And then the next situation, kind of going off of this scavenging note, was that, we had 3 different sharp-shinned hawk samples that all detected turkey DNA on them, and they were collected in late April and early May on separate days. So I'm pretty sure all of you here are very confident that that a sharp-shinned hawk, it's tiny, there's no way it's going to eat an adult turkey. So when I first saw this, I thought I don't know much about turkeys. Did it eat a turkey chick? And then, I looked it up, and, turkey chicks don't hatch in northern Minnesota, Wisconsin until early June. So a month after I collected these samples. And I talked to a friend who farms, and she says, even someone who raises free range turkeys isn't going to let their turkey chicks, roam in late April because they would freeze to death. So then I was well, there are turkey chicks that hatch in southern Texas and Florida in April. But it seems it was unlikely to me that a sharp-shinned hawk could be flying. It would take it about 3 days to make it at best from Texas to Duluth. And we know the gut retention time for raptors is about 6 to 8 hours. So, it eats something, 6 to 8 hours, it poops it out. So I really didn't think it was likely we were detecting any prey that was more than 24 hours old, especially considering like the low percentage of swabs that had prey detected on them. So that led me to the conclusion that these sharp-shinned hawks must have been scavenging turkeys, which made extra sense because turkey hunting season in Minnesota and Wisconsin goes from mid-April to the end of May. So that would be a really plentiful source of turkey guts and remains on the landscape for them. And this is doubly interesting because in my review of the scientific literature, I could only find one example or record of a possible scavenging event by a sharp-shinned hawk, and that was of a Sharpie caught in the Upper Peninsula of Michigan in the late 60s, and it had a porcupine claw in its foot. And the author suggested that it had been scavenging, but you can't necessarily say that because who knows, maybe it just swooped at the porcupine. So just like another example of like we've got sharp-shinned hawks, they're very secretive little birds. Maybe they're scavenging a lot more than we realize. And then this final little example here was that we saw this seasonal difference in prey detection in sharp-shinned hawks. So, I swabbed 141 sharp-shins in total, 63 in the spring and 78 in the fall. We had a higher percentage of prey detected in the spring than the fall. So I ran a chi-squared test to see if it was statistically significant, and my p-value came back at 0.057, which made me moan because if it had been less than that, we would have been happy and said it was significant, but it was kind of borderline significant. It's something interesting to think about that maybe, like sharp-shins are feeding more frequently during spring migration than fall migration. And it's something I think could use some more research done on it. All right, and then quickly, if you remember my second hypothesis, I thought that dietary DNA was going to maybe differ between species because of physical size and their mode of migration. We ran some generalized linear models to test for whether or not species, mode of migration, or physical size had an effect on the probability of detecting one or more prey OTU on a swab. And the long and short of it is that it didn't. The null model saying that there is no difference was the best model. So it was a great thought, but it's not what we found. But yeah, so in conclusion, it worked. I'm very happy that it worked. So DNA metabarcoding can be used to detect dietary DNA on colloidal swabs, but it's at a pretty low rate. So I thought I'd just talk a little bit about things that can be done to improve the method. First off, I think it's super important to conduct a controlled study. So that could answer us questions like, well, how much DNA, what's our chance of detecting DNA on a swab so many hours after raptor eats? Is there a real difference between these different species of raptors if we control what they're feeding and when we're swabbing them? There's a ton that could be done to refine the molecular methods. When I was talking about what barcodes we chose to focus on, I mentioned we picked ones that would target avian prey and mammalian prey. And as I'm sure you guys are thinking, you're like, you swabbed a bunch of kestrels and merlins and saw-whet owls, and they all eat arthropod DNA. And there are most certainly barcodes out there, primers we could have chosen that would have amplified, and we could have sequenced the arthropod DNA, but it just came down to money. We were able to pair the avian and mammalian barcodes in the same sequencing run. And if we had tried to do it with the arthropods too, we would have had to do a second sequencing run, and it would have been extremely expensive. So, there's things like that we could have done, but, maybe there is a better barcode locus on the genome that we could have chosen. And then, you know, as far as like things that we did in the field, like, you know, maybe if we, you know, collected multiple cloacal swabs per bird, we'd end up with more material. And then, you know, that would be more DNA to work with. I did cloacal swabs, but, you know, other people have been started to do DNA metabarcoding with buccal swabs, in nestlings and beak swabs, especially when you can physically see that there's blood on the beak, where, you know, prey remains stuck to the talons. And so maybe this is something like cloacal swabs could be used in combination with. And then I think there needs to be more like research done on like the feeding frequency of raptors during migration, because like for this method, which is already having a pretty low detection rate, I think it would be better to focus like resources on using like DNA metabarcoding for species that are feeding more frequently, just so that you have higher hits, and then think of other methods to use for those species that aren't feeding as frequently. And then another thing I thought about, of course, after I did my research, was this idea of factoring the time of day for when I collected the cloacal swabs, knowing that gut retention time is about 6 to 8 hours. So, you know, maybe swabbing a red-tailed hawk at, you know, 9 a.m. in the morning isn't the best time, because if it ate something at 8 a.m., like it's not made it through the GI tract. And if it ate something the night before, well, maybe it's already pooped it out and the cloaca doesn't have anything left in it. Same thing with owls. Maybe it would have been better for me to get up at 4 a.m. to swab saw-whet owls. So yeah, I'll just do a couple like pros and cons that I saw like with the DNA metabarcoding. Pros is that like with the cloacal swabs, you can use it on any species of raptor. It's relatively easy to collect the sample. You can use it any time of year, so you're getting away from this like breeding season bias. And then like there might be fewer biases to this method, but again, like whatever barcode you choose is going to have biases with it. Cons, we of course saw a low return of prey DNA on the swabs. It's very expensive. And then you need to capture the raptor. And I only put this in the cons because like it takes a lot of time and resources. And this project was only possible because Hawk Ridge has put the time and the resources into its banding operation. So if you were to want to go out on your own and do this, like it would be another like factor to consider. And then of course you have to take cloacal a lot. So yeah, that's my talk. You know, acknowledgements. We got grant funding for this project from the McCaney Fund, from the Bell Museum of Natural History. We got a grant from the Duluth Superior Area Community Foundation's Biodiversity Fund. Of course, Hawk Ridge was critical to this. The entire field crew, Frank from 2019, it's awesome. We did all our genetics work, molecular work for this project at the EPA in Cincinnati. Yeah, I think I mentioned this was part of my graduate work for UMD, and I had a lot of help and advice from all my graduate committee members. So with that, I think we've got quite a bit of time for questions if folks have them.
Excellent. Well, how are we doing? I don't think anybody put anything in the chat. Oh, but no questions anyway. Just a comment. I'm amazed at the amount of work you accomplished.
I thank you. Oh, we got a couple in the chat. Well, thank you for the comments.
Yeah.
Okay. So now
Here we go. Here we go.
So Sister Meg asked, do you think the ability to identify DNA will improve over time? So most certainly. Like one of the biggest drawbacks to identifying the prey species was that some of the prey species just weren't in the database. And so, you know, I'm sure if I go back in a few years and check the database, like, you know, we like a lot of those unidentified OTUs or the ones without perfect matches, like there will be a match in there. And then, yeah, just, you know, all the like advances in, you know, you step away from molecular biology for 2 years and it's like the field has changed, right? It moves so fast. High throughput sequencing, oh gosh, that started like in the around like 2010. So it really hasn't been around for that long. So a lot of the papers that started using dietary, like, you know, DNA metabarcoding for dietary studies were right around like, you know, started out 2010, 2015, you know, it's still relatively new and, you know, it's second generation, like what happens when we get third generation sequencing? All right, can you explain more about the capturing methods? So the raptors were all caught as part of the banding operation at Hawk Ridge. They use several different methods of nets with lures. So what we did was the birds were trapped normally. They were banded by the crew. And then like right before release, they would just hand the bird off to me. We'd take the swab and then we'd send them on the way. So yeah, I think, you know, for most raptors, it's about maybe 5 minutes of handling time, less for others, you know.
So you had a number of birds that you had sampled from, but they didn't land in the pot that had DNA in the cloacal swabs. If you could have your druthers, what would you want to get the results from that you didn't?
I would really like to have been able to use the arthropod like barcode, because I mean, we didn't get that many kestrels because, you know, they're kestrels, they're a little hard to trap. But, you know, like that could have been a reason why I just didn't get any hits for those species if they were primarily eating dragonflies in, you know, early September. I think the sawaddles I thought too would be really interesting to have with the arthropod ones, because, you know, they do eat, you know, moths and stuff. But then again, I was also like collecting those samples in October. So maybe that's a really great example of like seasonal differences in diet, because there's just not as many arthropods flying around after the first hard frost. And then again, I also just wish I had been able to get out to collect more sawaddle samples during that fall. I was also teaching that semester, so I had to be in class at 8 a.m. So that put a little damper on that. Yeah. Oh, and then Patty, to like add on to your question about capture methods. So yes, some of the nets are mist nets, but they just have a larger mesh size. And then there's also nets called the Gaza nets, which are a net where when the hawk or falcon hits, it kind of like drops down around. So it's like a quick release net. So it doesn't have the same pockets as a mist net. And then there's also bow nets, which is a net that's on the ground and the raptor lands, and you have a physical button that you push or a string that you pull that releases the net over the top. So it's kind of like, not like a cage, but similar to that. So then all you have to go do is run out, grab it, pull the net back. Oh gosh, let's see. What will you be working on next? Well, I'm not currently involved in any research projects, but I'm doing an AmeriCorps service where I'm doing a lot of like ecological restoration training. And I think, you know, I talked a little bit earlier about like the implications of dietary studies to land management. So as far as where I'm kind of trending, I think that in that general direction of like using research to make like sound land management decisions, restoration projects, that kind of thing. Yeah. And I think it'd be really interesting too to see like how like diet would vary, you know, within a species between different habitat types or in the same location over time as like habitat changes. But I have no immediate research. I don't have any proposals drawn up. Will my research be used in conservation work? I think of my work as like a small little piece in the big umbrella of conservation. And that's what most science is we all do our little chip, and then someone can hopefully think to use colloidal swabbing on a different project. You know, like the results I found were interesting, but you know, it just wasn't like a large enough scale thing to be like, oh, we now know so much more about like the diet of sharp-shinned hawks. It's like, this is a tool you can use to do more studies.
So Lisa didn't tell you, but one of the things that she found out, she did mention that many of the birds they didn't have DNA samples for. And I know we worked really hard on expanding the DNA sample set.
Mm-hmm.
Did that actually happen, or are they still sitting in a freezer somewhere?
That's a great question, because we were starting that collection right as I was finishing my master's. So I'm not sure where we're at on those, if the guys at the EPA were able to like run those sequences or not. But they're certainly still there. They haven't gone anywhere. But yeah, a lot of the warblers, fall warblers, were, you know, missing from the database too. So.
Sounds like we've got another question coming.
Or
a comment anyway, compliment.
Thank you.
Lisa, while I'm thinking about it, make me the host again so I don't forget.
Gotcha. All right.
Thank you. Thank you.
Will do that.
Cool. Anybody else got questions for Lisa? Very fun stuff. I love telling the wild turkey Egyptian story. That's just one of my faves.
I have the photo of Emily there holding the sharp-shin, because the original slides from this all came from my thesis presentation, which was being delivered to a lecture hall that was a mix of general biology undergrads and professors who do research in the medical school. So I had to first go over what is a raptor and what is hawkridge. So it's nice to talk to people who are more familiar, but that wouldn't have made sense to any of those undergraduate students if they didn't really understand how small a sharp-shinned hawk is. Yeah.
Cool. Well, anybody else? I'm excited to have heard the whole thing and really, really hope that people continue to do some work on the migratory diets of these birds, because I think it's going to tell us a lot that we don't know. So Yahoo for starting the trend.
Well, thank you.
Everybody's so quiet tonight.
It's hard to know what questions to ask for. Yeah. It's not an easy. It's very interesting.
Yes, I know. I threw a lot of information at you guys really fast. We got a question from Allison about if the recording will be sent to the registrants. Where are you going to post the recording, Margie?
Yeah, I will post it on the, I'll post the link to it on the Hawkridge Pick a Bird Brain series spot. And if I remember, I'll send everybody out a notice too that I did it. But if I don't, give me a couple of weeks and I will get the video up on the link on the website. So, yay.
You can edit out every time I stumbled from the recording.
You didn't. I didn't notice any anyway. She's done this presentation a few times.
I bet.
Oh, well done. Well, anybody else gonna chime in here? Otherwise, we'll turn you loose to enjoy the rest of this gorgeous spring for the moment night.
For the moment, yeah.
Well, thank you guys for all coming. It's always fun to be able to talk about the work we did together at Half Ridge.
Yeah, thanks so much, Lisa. We
Yeah.
Yeah,
really
thank you.
appreciate it.
Bye.
Yeah, very interesting.
All right.
Yeah, thank you. Goodbye.